Good day, Junior Techies. Welcome to this video where we're going to be looking at a couple of bonus questions. This is particularly going to be looking at bonus question 10 for our exam preparation. My name is Mr. P. Thanks so much for joining us here. We're going to be working on a bit of exam technique and then also a couple of past paper questions that I'm sure you may have seen before, or that you're going to get a little bit of experience at answering in your preparations now. So let's have a look at what we have here. This is question 10, as I said, and the diagram below shows crossing over during meiosis. You're going to see many questions dealing with crossing over because it's one of the most important concepts when we look at meiosis as well. And you'll see here that a bit of genetics has also been thrown in because we can see the alleles represented on these homologous chromosomes during the phase that we see here. So for one, we can see that crossing over is taking place. So the first thing for us to do is to identify what phase this is. And we can see clearly that it's prophase 1, because prophase 1, we have the crossing over happening. So getting context here, we can assume that we're probably going to be getting the answers to a couple of the questions already. And we can see that when we look at getting context here, we see that we have these two chromatids represented as X and Y. So important for us not to get confused here between the gonosomes and then the simple labeling of these chromatids. So these are definitely not gonosomes. These are just letters X and Y that are referring to each of these chromatids that are joined at the centromere over there. So question 10.1, let's see. We have named the phase of meiosis during which the process represented above takes place. So this is quite simply prophase, as we identified while looking for our context. Okay, so now let's have a look at question 10.2. So now it gets a little bit more tricky because it says to us here we need to describe the process of crossing over. And I've taken a snippet of the memorandum here because it's so important for us to get this in its sequence correctly. So we've got homologous chromosomes where the chromatids of each overlap at the points called the chiasma. So that's what we have represented by this point over there. There's our chiasma. And then at that point, genetic information is exchanged. So we can expect that these two bivalent or homologous chromosomes are going to look a little bit different after the exchange of genetic material, which has taken place at the chiasma there. So there's our answer answer for our describe question. Because if we move on to question 10.3, we can see that the next question here is going to be looking at explain. And I've quickly popped in the memorandum answer here too, because now it's the what and the why. So describe is the what is happening, and explain refers to the what and the why of what is happening. So let's see. We've got the importance of crossing over being that it introduces genetic variation, leading to new characteristics which may be favorable or unfavorable. Obviously, we would like the favorable characteristics to be present in the offspring because then our chances of survival would be better. But if unfavorable characteristics are the result of this crossing over, it may sort of not be beneficial when it comes to natural selection, because through environmental changes, organisms are either more suited to their environment or less suited depending on their characteristic traits or their phenotypes. So ultimately, looking at the crossing over, it adds to, or it does increase genetic variation. Whether it be favorable or unfavorable, it still increases the genetic variation in that species through the offspring. So that's our explain question for 2 marks there as well. Okay, so highlighting a few points here not to forget. I think we definitely get our 2 marks there. Now looking at question 10.4 very quickly, it says draw a diagram giving the position of the alleles to show the structure of chromatid Y after crossing over. So now we've got to go back to our diagram. This is also for 2 marks. So let's see what our diagram tells us. So we know that we're looking at chromatid Y. So we know that chromatid Y has a specific structure, and we have the centromere over there. It's only 1/2 of this chromosome which is still joined at the centromere. So we're only going to be doing the chromatid Y. Now the position of the alleles we need to put in. So we can see that D and R are not affected by the crossing over. But the last allele, where we've got either our dominant or our recessive B, our capital or our lowercase b, we need to now determine if crossing over has taken place here, what will the chromatid Y look like? Would it have the dominant B or the recessive B allele? And in this case, we're looking at B. But we do need to represent the color as well, so we can show that the crossing over has taken place. So this is just me with a pencil on the side of my page, making a couple of notes of what I think I should be doing as my answer, and then I'm going to take that and I'm going to actually put it in as my answer to the question here, 10.4. Okay, junior techies, well, I hope this has helped a little bit in analysis of a question. Don't let questions like this intimidate you. Get context first, then look at the questions. You'll find they're not as difficult as you think they are. And I do hope that in visiting these videos and practicing these questions, that you do get more confident with answering these questions and that you do well in your final exam. Well, thanks again for joining me. I'm Mr. P. I do hope to see you again in some of our future videos.